NTU World of Wisdom. Welcome to High Impact Thesis. In this podcast, we speak with researchers from various scientific fields to talk about the motivation, goal, and potential impact of their research. We also want to give you a sense of how a Ph.D. is carried out with an emphasis on the PH, the philosophical aspects involved in pursuing a Ph.D.
So, Sweta, welcome to the first episode.
Hi everyone, my name is Sweta. This is my final year, hopefully, as a PhD student at NTU. I came into work here more or less in an interdisciplinary domain between pharmaceutical sciences and maybe even biomedical sciences. So the core of my research is based in polymer chemistry, but the overarching, I would say the application would be in drug delivery, and more importantly, centered around pulmonary drug delivery. I do have a model drug that I'm working with, which is targeted towards treating Parkinson's disease, but we would like to hope that this mode of delivery would be applicable to multiple drugs and hence to multiple diseases.
All right, I see. So this is a broad description. So can you tell us maybe more description about what drug delivery means? Why is it a challenge in general?
Actually, drug delivery can be in many different ways. It literally can be the most old form, which is the oral and the topical one, which has been there since, I would say, age immemorial, to even the more newer technologies, which is transdermal, through microneedles, even the kind of work that I do, which is through the pulmonary delivery, which is a combination of inhalation as well as both through inhalers as well as nasal depositions. So there are many avenues, and each of these avenues have their own challenges as well as benefits. And exploring each of these avenues actually requires different approaches. And I don't think I have enough knowledge in the other domains, but I think for the kind of route I'm trying to explore, which is the pulmonary one, has several, as I said, advantages and disadvantages. If you want, I can go into details for it over that.
Yeah, we can go into
sure.
that
We're gonna get into details
at some point.
Sure.
So in general, what would you say like the motivation? Why did you choose this area of research in particular? If you had,
That's the funny bit. I think this work kind of chose me. So when I was doing my master's in India, I was doing in a completely different topic. I was working with heavy metal toxicity, and I was working with two elements primarily, which was the arsenic pollution and the fluoride pollution in different areas of West Bengal, and more focused on the mitigation of it. So I can primarily say that I was working on the chemistry of it. And then when I was looking through various projects at NTU, I came across this one, and I realized that this area does attract me because it has a lot to do with chemistry, but in a different domain. By the time I was finishing my master's, I had already worked with heavy metal toxicity for like 3 years, both as my bachelor's thesis as well as my master's thesis. And I realized that okay, maybe there was not and much beyond at that point I could have done, and hence I started to look at other domains, and this domain found me because it had to do a lot with chemistry, and a lot more with chemistry than other people actually give it thought. And then I spoke to my professor, who was in fact a large motivation as to why I'm here. He explained what kind of work he does. He explained the kind of scope I have worked with to this work, and I realized that this is an area that I can definitely make an impact in if it amounts to something that's worth doing. And that's exactly why I'm here exploring that possibility.
I see.
Okay, I want to step back just a little bit. You when you started, you mentioned polymer chemistry.
Yes.
Right. So can you just give us a brief? What does that mean?
Okay, so I think in chemistry in general, right now we can broadly say, okay, there is physical chemistry, there's organic chemistry, and then there is inorganic chemistry. And in organic chemistry, one of the largest fields of study right now is polymer chemistry. It can be from industries, which produces products that we use daily, to pharmaceuticals, where they are utilizing these specialized synthesized polymers and copolymers and block polymers to create these micro nanoparticles, which are usually the vehicles or vesicles or whatever form you're generating them into as a loading material, as like sort of like a, how do I say, as
Like
a
a
component,
vehicle?
yeah, like a
Okay.
vehicle which allows you to carry drugs into various systems. And it is in one of these domains that polymers over the last few years, maybe even the decade, have gained so much attention that I would like to say that it's a separate field which can be called the pharmaceutical polymer chemistry.
I mean, because when I hear polymer, I just think of plastic. I don't know
Plastic,
why. It's just.
I know, so did I.
Yeah.
I did too. I mean, when I came in, I was like, okay, there's chemistry, and there's organic chemistry, and there's physical chemistry. That's it. Those are the divisions, and everything else belongs into their, subsets. And then I came in, and then, of course, we all do our literature review.
Right.
And there's this vast ocean of, information where people have been using these. And it's so fascinating because some of these have even gone on to obtain FDA approval to be used, and they are very successful. So people have, of course, taken this a bit further and now trying to explore if these can indeed become the future excipients for various kinds of drugs. Some of the traditional excipients. So excipients are components onto which we load the drugs to give them volume or to help ensure that their properties are not lost. And then these are administered, any form, oral, topical,
Right.
ingested.
The closest thing, at least in my experience, is the capsule tablets. Right? Is that like was that an early stage of research in this area? Like,
The tablet
Capsule.
coating, you
Yeah.
mean?
Yeah.
That of the
container that
capsules?
you can actually ingest.
I'm not sure if those are technically made of polymers because those have been around for quite a number of decades.
Right.
And I'm not sure if this is the one, like if polymer were one of the components they were using, because I'm not an expert and I cannot claim knowledge
I mean,
in
you
that
just said
domain.
it's a big.
It's a big
big
domain.
field,
Yeah.
So is this work primarily concerned about using certain molecules or polymers as a carriers of drugs?
Yes.
Or as, like, is there any possibility for targeting certain tissues within the body? Or
Of course. I mean, it really depends on how you're administering it, or where you're sending your microparticles, like how these people are taking it in. In my case, they will be inhaling it through a dry powder inhaler. So the first environment they'll be meeting are the lungs. And once they're in the lungs, we would think that these microcarriers would allow the drug to diffuse out and in sort of allow them to, how do I say, mix or rather be absorbed into the blood system, where they will be carried to the brain across the blood-brain barrier, where they will have the storehouses for dopamine, which is, okay, so before I talk about dopamine, why I'm talking about dopamine, I work with a drug which is known as levodopa. Now, levodopa is the gold standard for treating a disease called Parkinson's disease. And one of the main features in Parkinson's disease is that over a long period of time, these patients start losing their dopaminergic neurons, or neurons which produce dopamine. As a result of which, they suffer from a multitude of symptoms. They can be a loss of control over voluntary movements to even dementia, depending upon the stage they are at. So in such a scenario, one of the main ways of treating it is to supply the body with the dopamine. And this administration ideally has been done through the ages through the oral route. Now, there are certain challenges of the oral route, which is why we want to bypass that system and target the pulmonary system, which gives a sort of benefits over this oral route. And that is why when we administer the dopamine or levodopa, the pro-drug form of dopamine, we would assume that once it reaches the lungs, it will then be dissolving into or absorbed into the circulatory system, where it will be carried across the blood-brain barrier, which is where it will be transformed into dopamine and stored in the various storehouses that we have for the body to be using it later.
So it's a precursor to dopamine, not?
Yes.
Oh, okay.
And usually levodopa is administered in combination with another drug, which is known as carbidopa. And carbidopa is sort of like a decarboxylase inhibitor. So in our body, we have an enzyme called decarboxylase, which is notorious for breaking down levodopa within the circulatory system. So if we administer it with tandem with carbidopa, we ensure that levodopa is not broken down while it's in the systemic circulation. Now, this carbidopa cannot cross the blood-brain barrier. So when it reaches the blood-brain barrier, it is only levodopa which can cross it, which is where it gets transformed into dopamine and hence utilized.
I see.
We want to get into what you're actually doing in the lab, like your day-to-day activities, and mainly what are the goals that you have set. Basically, your thesis, right? I'm sure you're working towards a certain target.
Of course. I think so. In 2017, when I came in, my supervisor gave me this blank problem statement, and he said, I want you to work on pulmonary drug delivery to improve it in some way with levodopa as your model drug, so that we can improve the way Parkinson's is currently being treated. Now, there are several ways in which you can look at this problem, right? You can look at the formulation, you can look at the delivery itself, like the medium in which it is being delivered, or you can look at the disease. And that was a choice that was up to me. And I chose to look at the formulation. So my thesis, if I had to sum up in a sentence, would be on developing different kinds of formulation with levodopa as my model drug to see which amongst these work the best in various scenarios. Now, the different scenarios that we need to consider is stability. Since I am delivering it through the pulmonary drugs, like the pulmonary system, I need to ensure that a certain quantity of the drug is indeed reaching the system. And the third thing that I need to ensure is that once reaching the system, this drug remains stable enough for it to be absorbed into the circulatory system. And the fourth thing is I need this system to be sustainable over a 24-hour period. So these are the four main targets of my work.
Okay.
And these are all interlinked. So once the formulation stage is done, and then I explore how well these are distributing through the lungs, then I explore the stability, and for if it is sustained, this stability is sustained over a 24-hour period.
I see.
Okay. I'm kind of I'm kind of interested. Like, how do you actually monitor these different stages? Like, you mentioned like the drug reaches a certain place in the body, and then it moves to the next part where like there's a separation of the chemicals, right? So how do you actually are able to tell what's happening inside?
So that's actually a good question, because that was something I was pondering about a lot initially. But I think that there are enough in vitro studies which are available, as are characterization methods that tell you whether your drug is whether your formulation, is the optimal formulation for your drug or not. For example, once formulated, that's the straight and simple method. And I use the spray drying technique to do the formulation. And this why I chose spray drying above other methods is because it's already there, And
Okay.
if something is already there and it's been working very well in the pharmaceutical sector, it's more easily adoptable.
Right.
People will be more open to that method. So that's why we started with the spray drying. The other benefit to this method I found out was, since I'm looking at a powdered formulation, there is very few method that comes to give as much efficiency as that of a spray dried formulation. It really gives you the powdery form you're looking at. The next thing you would have to look at is whether these are really the ones that you would need for pulmonary drug delivery. Because for pulmonary drug delivery, especially if I'm using a dry powder inhaler, I am looking at a formulation whose microparticles need to range within a certain therapeutic limit. There's between 1 to 10 micrometers.
Oh.
Anything less than 1 micrometer, your body is just going to breathe it right out. Okay? And anything more than 10 micrometer, it's not going to reach your lungs very well. It's just going to get stuck at your throat. So you're looking at that ideal therapeutic range. So that's one of the biggest criteria when you're checking whether your microparticles are good enough. The second way to establish whether they are is to run various mechanical simulations or experiments, I would like to say. And we can do this with instruments such as the Anderson cascade impactor, which mimics the human lung. A mechanical, it's a mechanical human lung.
I just wanted to ask you about that. How would you model the environment of the lungs inside?
Yeah, so we do that in two ways. One, we look at it from a mechanical point of view, and the other, we look at it from a biochemical point of view. So the mechanical point of view would be the use of, as I said, the ACI or the Anderson cascade impactor or the next generation impactor, because these sort of form like a sieving mechanism. Okay, so they have different stages with different pore sizes. And once you deploy your particles as you would breathe in through a dry powder inhaler, your particles would settle down because of gravity in form of their particle size. And if you recover from each stage and you analyze it back, you will know what is the fraction of your particles which belong to each category in terms of size, and then you know whether this will work or not, because you can express the percentage in form of two things. One is the fine particle fraction, and the other one is the emitted dose. So greater the values for these, the better is your formulation. So you know. So that's one mechanical characterization that tells you.
Then the second one is biochemical, right?
Yes.
That I would imagine would be much harder to...
Oh, yes. It's the most troublesome one. So over here, when we did the biochemical analysis, we were actually trying to establish two more targets. The one was the stability over a 24-hour period, and the second one was to see if indeed our drug was leaching out of the polymers onto which they were loaded, because that is one of the main concerns. Now, ideally, people use polymers because they want them to have a sustained release over a very long period. Now, mine is somewhere in between. I do not want these microparticles to last for days. I want them only for 24 hours, which is like trying to hit that sweet spot, which is very impossible to achieve. So that was something we had to sort of constantly reiterate our formulations a little bit to check if we are actually getting to the 24 hours. How we do that is we use an in vitro testing method. It is called the drug dissolution study, and it's a very common technique in the pharmaceutical studies. And here you can, so the medium in which you would explore also varies depending upon which region you're targeting. I use something called stimulated lung fluids. So stimulated lung fluids are solutions, artificial solutions, which mimic the environment inside your lungs. And there are two kinds of stimulated lung fluids. One is a Gamble solution, which mimics the interstitial lung fluid, and the other one is the artificial lysosomal fluid, which mimics sort of the internal environment of something called the alveolar macrophages.
I see.
Very interesting, because these are the ones who are responsible for sort of phagocytosing, or I would say ingestion of foreign particles within the lungs. So these are sort of like defense responses by the body, where they take in the foreign particle and they try to break it down. So that's the second environment that my particles will be, interacting with.
I see.
So it's very important for me to know how they are faring in just the lungs and then these macrophages.
It sounds like a lot of variables.
Yeah,
involved.
very hard to control. But I would imagine these procedures, like for mimicking the biochemical environment of the lungs, are standard, right?
Yeah. So these two solutions are standardized. They have been used in environmental studies as well, especially air pollution, where a lot of the particles go into your lungs and the scientists want to study how it's behaving and how it's acting. So this is like an established dissolution medium.
So I'd imagine there are certain advantages for one test, like the mechanical test, and there are other advantages for the biochemical. That's why you're using both of them?
Yes.
Okay.
I use both of them. So for the mechanical one would be to ensure that I'm getting the correct particle size. And the biochemical would be to ensure that I have the chemically right formula. And the biochemical one is to ensure that we're getting the chemically correct formulation. So it is like a two-prong thing that you have to do.
Okay. We haven't actually got to what you do, like physically, other than using these tests, right? Like what actually happens when you are there in the lab? Because I know that you're there for a long time, like the whole day, every day.
Would you be surprised if I tell you that my dissolution study literally lasts 24 hours?
That's what confuses me. Like, how can an experiment, like, I understand for certain, it's mainly because I don't really understand what you do, so
when
I'm sure it makes sense, right?
So for example, if I'm doing my fabrication, it won't last long. It'll take me about 2 to 3 hours.
Is that the formulation
Part?
Okay.
Yes. So 2 to 3 hours from when I start prepping my reagents and my material to the collection of the material. Then once you've collected these materials, they need to be tested out, right? So you have to do particle characterization, and that involves visually seeing them, which we do with a microscope, like for example, the scanning electron microscope. And then once you know how your particles look like and you understand that these are the ones you want to move ahead with, then you start the solid state characterization, and you do it with the help of things like X-ray diffraction, the FTIR, which sort of tells you a little bit more about the various components and how they're behaving or interacting with one another. Then the other thing that I do use is called the DSC, which is a differential scanning calorimetry. It tells me how my particles are degrading in presence of heat. It's important because one of the other things you need to ensure about your formulations is, since these are pharmaceuticals, they need to have a decent amount of shelf life. They can't just degrade because of heat or some other factors.
I see.
So you need to validate these points, that they survive through this, basic temperature range, basic pH, and those things. Also, of course, one of the biggest studies I spend most of my time with is the dissolution study, because it runs 24 hours. So there's one whole day where I'm awake, and then there's the next day where I'm sleeping. And then I have to do these in like multiple times to ensure that, I can replicate the data. And it's an evolving process.
So what's taking place exactly during these 24 hours?
During these 24 hours, I will be I'll be drawing samples out once from the experimental vessel being my stimulated fluids with the microparticles in it. And I'll be checking for the drug concentration every hour to see how it's evolving with time. And so it varies. This thing varies for other people, but for me, I chose 24 hours. So I need data from every hour to see if it's running correct.
So you're doing
every hour?
Every hour.
24
Like you don't
hours.
sleep
Yes.
for 24 hours?
No.
Wow.
So previously my supervisor had the same reaction. He was like, no, break it up, do 12 hours and then do 12 hours. But I thought it leaves a lot of room for error, mathematical error. and it does not give good results. So I rather do it at one go, see how it
Gotcha.
works, and then sleep for the next 24 hours.
I see. But I would imagine that most of the change would happen within the first few hours.
A lot
Is
of
that
the
correct?
drug, yes, a lot of the drug actually leaches out within the first 6 hours.
Yeah.
And then it becomes quite steady.
Mm-hmm.
And this is something we are actually looking at, because when we talk about Parkinson's, these are people who have a very poor quality of life when they are in their off phases, that is when their body do not have enough dopamine. So you would need a drug that would act immediately, okay? At least give them the immediate shot, just for the body to adjust itself back. And then if it slows down over a 24-hour period, it's not a bad thing. We will take that. But initially, they would need that certain hit for the body to,
Okay.
come back to.
But why did you
normalcy.
mention earlier that you do not want your drug to stay for more than 24 hours? Like, what's the issue?
Okay, so over here you need to know a little bit of a background then. So right now, as I said, levodopa is the gold standard. And the most common way in which levodopa is still being used is through oral delivery. Now, the oral delivery, because of so in our stomach, we have different kinds of gastric enzymes, right?
Yeah.
And these gastric enzymes break down the drug very fast. So studies have estimated that about only 30% of the ingested drug reaches the blood, actually manages to cross the blood-brain barrier and is effective. 70% is lost. As a result of which, these people have to be dosed at a higher percentage to ensure that a greater amount of drug is reaching the system. And that itself has several side effects. Moreover, since these drugs are being ingested, they need an hour or two before they can reach their peak therapeutic activity. Like at least the peak plasma concentration will not be attained before an hour or two. So during that period, they still are not doing well, right? Moreover, the other problem is, as the disease progresses, one of the main symptoms that arises due to loss of voluntary control is your ability to swallow. So you cannot swallow very well. So if I'm looking at a patient who is in an advanced stage, probably requires more than 2 or 3 tablets a day to treat his symptoms, he would need to swallow these tablets. And he has a swallowing disorder. So it's difficult physically, and after a while, I'm sure it has a psychological thing coming in where he would be like, no, it's okay, I don't want to do this. And they would skip their dosage. So because of these factors, we thought that the pulmonary drug delivery will be a little bit easier, because with dry powder inhaler, you just have to push a button and it's in, like we do in the asthma drug, right? And it has been proven that pulmonary drug delivery allows the therapeutic plasma concentration to peak within 30 minutes.
Oh, that's
That's
really
fast.
Yeah.
That's really fast. So that's one of the reasons why we went for the pulmonary drug delivery. But now why 24 hours? The main thing is, since it peaks so fast, we know for a fact it cannot remain sustained over more than 24 hours. Because most of the drug will be released within that time period.
Okay.
So to ensure that it is delayed further is counterproductive to our first target, which is to ensure that they have a quick hit when they need it. So that's why we're not looking at a sustained drug delivery for more than 24 hours. So for 24 hours, they don't need to take 4 pills a day. They can just take one inhaled, one inhalation or one puff. But beyond 24 hours, we are countering the immediate need within the first 30 minutes that this method is providing.
I see. So I'd like to move to another point, which is about what are the biggest challenges or barriers to pushing the development further in this area?
I think it's firstly Parkinson's, although it affects many people, it's still a very niche I would say, a very niche disease, right? In Singapore, I think it's just 3 in 1,000, every 1,000 people above the age of 50. And this was a survey that was conducted by Ministry of Health, I think, and it came out in one of their guidelines in 2004 or 2007. Okay, and there they said like only 3% above the age of 50 suffer from Parkinson's disease. And since it's so niche, the amount of research that is happening is, of course, not as wide because it doesn't impact a large population. Secondly, we're looking at a geriatric population. They may or may not be open to a new mode of drug delivery, right? It can also depend upon training. Like these people, they don't have control over their movement. So sometimes they would need assistance in order to ensure that they're taking the drug properly. Or they have enough inspirational volume, like breathing volume, to be able to breathe in the entire amount of the drug as required. Fourth would be not a lot of people are, as I said, it's still the oral method that a lot of people are concentrating on because it's easier, I would say, and hence not a lot of people are exploring this. But yet at the same time, there are studies that are going on simply because they think that this is a possibility that should not be left unexplored. And because of that, we do have an FDA-approved drug, which is called CVT-301, which does pulmonary drug delivery of levodopa. And is either under clinical trials, but towards the end stage, so it should be in the market soon, and it's a promising sign that, maybe the world is opening up to alternative routes.
That's good news, actually.
I once heard about like some people are trying to develop something similar with insulin for people who are suffering with.
They have actually.
Because taking shots every day are very
for diabetes.
Diabetes, for insulin.
They definitely have for insulin. I think they even used something called a polaxomer, which helped insulin because insulin is a hormone and it requires a specialized encapsulation, if I'm not wrong. And they use something called polaxomers, which are hydrophilic polymers, to ensure that these two are, these
compatible. Maybe?
Yeah, I think so. But it's very new, I think. 2010-ish, I would say. I remember reading it very vaguely about it when I was researching for my work.
So nothing on the market yet?
I have no comments on that.
Wait, so what's the name of the drug?
CVT-301.
That's for the insulin, though.
No, that's for levodopa.
Oh, levodopa. What is levodopa?
Levodopa is the levo form of dopamine. It's a precursor of dopamine.
And so when you're formalizing the drug, like the
like the entire formulation of the drug.
You call it formulation, but what is it? Is it the chemical product?
So the formulation is both the drug and the excipients. And these excipients contain, should I say it? It's a secret.
I mean, if it's a secret.
So the excipients contain, I'm going to broadly say it, a polymer, a carbohydrate, and 2 other additives, which ensures the property of the drug is not hampered or degraded.
Right. So my question was actually regarding the excipients. Like, is it possible to use this formulation, the excipient, for other drug delivery?
That would be the ideal case. So if once it works for levodopa and we know for sure it does, we would like to apply at least the same excipients for similar drugs whose chemical properties are similar to that of levodopa's, and then see if it works for them first or not, and then maybe move on. But I'm sure I have tweaked this formulation about 20 different times because I know for a fact I have 35 different Excel sheets with 35 different formulations on it. So it goes under a lot of tweaking here and there, depending upon what the results say. So there will definitely need some tweaking. But the idea is to come up with at least one system that works and it can form your foundation for other systems to be engineered later on.
So it's kind of like once you do it once, then it's much easier to replicate the same process with other drugs.
Yes. Wouldn't say replicate, but to design similar.
Similar.
Because replicate is ambitious.
So it has to be customized to each drug, basically.
Yes.
Process.
The ratios in which we are using the various excipients, the conditions in which we are preparing it. Because, for example, levodopa is quite stable at 80 degrees of spray drying, which is the temperature I use. But other drugs may not be so stable at that temperature,
Stability,
right? Some drugs even need much, much lower temperature for them to work, like for them to remain in a stabilized condition. So in such a scenario, spray drying may not even work. Right?
Yeah,
so I might even have to change the way in which I am producing it.
Right.
But my excipients might remain the same.
All right. So we did talk a lot about your research, right? So the background, the work you do, and like where can this go from here? So in addition to all of that, we also wanted to touch on your personal life as a PhD student, right? You're away from home in a foreign country, but still a lovely place, Singapore, NTU. So what keeps you, what do you do for fun? Like, what do you do to engage with
outside your research?
Yeah, exactly. Engage with other PhD students.
Actually, I was going to say, what have I not done? I think...
It sounds like you've done a lot then.
I would like to hope so, because I think that one of the main things that you need to do as a student, because I'm lucky enough to still call myself a student, is that you need to explore the surroundings around you, and you can do it with the help of people around you and the various situations you have. And one of the main things that I learned while doing a PhD is it's a simultaneously lonely journey, which can also be very fulfilling if there are other people involved. And why I say simultaneous is because there are a lot of other people who are involved, and it's up to you how you drag them in your net. Right? You need to reach out first and forge these connections because you need them. You would need them professionally because you're not going to be good at all the instruments, and you would need help with other people and their expertise. You would need them personally, maybe just to rant about the experiment that's not working out for the last 2 months. and you would also need them like socially, because everybody loves a cool beer at the end of a very long day, right? So there are different contexts in which you would need these people. And of course, sometimes when you're doing something constructive that you can do on campus over here, and I'm sure you can do it on other campuses as well as a PhD student, like engage with student body community, engage with the community in general. So many ways in which you can give back while you get something in return as well. So I think it's up to you as it's just who you are as a person and how much you're willing to put yourself out there and hope that you'll get back something in return.
That's great. So, like, of course, this podcast being an NTU WOW initiative, NTU WOW's main initiative is, as you know, TEDx NTU. And you've been involved with TEDx for how long now? For 4 years almost?
I think I'm old. 4 years. 4 years.
That's almost your whole PhD journey.
Yes.
With the TEDx.
I'm so glad I answered that random email that popped up in my inbox, in October of 2017. And I had this amazing chance to just come up on team. On the first year itself, I was just impressed. Like, here is a body of what? That year was about 100 volunteers. This body of 100 volunteers made an event happen, which involved 1,600 people.
Yes, that is the full.
Yeah, I mean, I think
auditorium.
that was. Yeah, exactly. I mean, it's just when you're there, and you see the evolution of the process, where a nascent idea, which a speaker has, and the speaker is not even sure of, is curated by this amazing team with insights from, say, 10 people converging and then, coming together so harmoniously. And then on the D-Day, you have this speech or this talk ready, and the speaker goes up on stage and does his or her best. And you feel that you've raised this child together as a village. It's an amazing feeling. And I've been fortunate enough to have felt that for the last 4 years.
Right.
And it's very addictive.
I'm jealous now.
It's extremely addictive.
Right. Yeah, definitely. It's an amazing process. So yeah, since I've also been involved, I can.
Yeah, I think I met you through TEDx.
Right, we did met
you,
through
right?
through TEDx and you, yeah.
So it's great.
But yeah, I did have I did want to talk about like your love for cooking and your love for reading and all those things.
Ask away. I'm yours for the evening.
So what got you, for example, to reading? You do seem to like to read a lot.
Okay. I think I loved reading even as a child. When I was a kid, I remember my parents taking me to the book fair. We have this wonderful book fair back in Calcutta that arrives every winter. And it's a paradise if you are a kid who loves to read. You're just there and your mom's like, you can take whatever book you want and we're paying for it. I'm like, okay, you will regret
The
this.
nerdy kid candy store.
Yes. So yeah, I think that's where I started reading. But I think one of the main things that helped foster this love is I had never faced any kind of censorship growing up. So I was allowed to read anything and everything. So my parents would not take away a book because it's too old for me. They'd be like, you can judge for yourself if you want to continue reading this book or not. And that was backed by my granddad, who was a professor at a college back home, and he of course had a huge library where he was reading books about skeletons, and he had animal skeletons, and he would he would be laying down the skeleton of a guinea pig on the table, open the book, and he'd be like, this is this bone, this is what it's called. And to a 7-year-old, that's like, wow, like the epitome of coolness, right? So yeah, I've always been surrounded by books, automatically translated into my thirst for just reading anything and everything, as long as it interested me. But I could never finish War and Peace, though.
War and Peace.
Could not finish that book. I tried.
Okay. It took me, I had similar experience with the 100 Years of Salt. It took me like 4 tries.
Oh yeah, that was
book.
a hard book to read.
Right.
But it's so well renowned by people, right?
Yeah.
Everybody loves it.
Yeah, it's a great book.
So yeah, thank you very much for coming, and thank you for giving us your time, and wish you all the best.
Thank you so much for having me here. This was a lot of fun.
You're most welcome. Thank you.
Thank you.